My name is James Pelletier. I'm a postdoc in the group of Saul Ares at the National Biotechnology Center in Madrid, and appreciative for the opportunity to share this work on cell division in Syn3A and would much appreciate your feedback. So the first part, I'll be presenting on behalf of this collaboration between a team at the JCVI led by John, Elizabeth Strakowski at NIST, and co-authors at the MIT Center for Bits and Atoms and UC San Diego. So for context, while JCVI Syn 1.0 provides a benchmark for normal cellular size and shape similar to that of wild-type Mycoplasma mycoides, Syn 3.0 exhibits extreme variation in cellular size and shape, where some cells are greater than several microns in diameter, and the cells are largely stuck together into these large clusters of cells. So this suggests some problem with cell division. And we're interested in cell division for both fundamental and applied reasons. Fundamentally, cell division is central to cellular life. So in this genomically minimal cell, what genes are required to regulate the process of cell division? And then from an applied point of view, many other aspects of physiology depend on cellular size and shape. So it's advantageous to have a cell that has lower variation in cell size and shape. So one could say for a model, know what volume is the cell or how many molecules are there inside of the cell. So these motivations led to the identification of JCVI Syn3A, which has a nearly minimal genome that retains an additional 19 genes that are not retained in Syn 3.0, but it more resembles Syn 1.0 in terms of its size and shape. So those 19 genes are organized into 8 clusters that likely represent transcriptional units based on the work of Troy Breyer and colleagues at the University of Illinois. And the first cluster that caught our eye was a cluster that contains FTSZ, which is a bacterial homologue of tubulin. It can form filaments that often localize at the division site. It's highly conserved in bacteria, and it can also generate forces that can constrict the cell, such as in this example, which is from an electron cryotomograph of purified FTSZ inside of a liposome. And in bacteria, FTSZ will interact with many other proteins involved in cell division. So given its known role in cell division, our first hypothesis was that FTSZ, if we put this back in, it would restore normal cell division, but this hypothesis was wrong by far. The cells still exhibited extreme variation in cellular size and shape. And looking at what was known about the functions of the other genes, there weren't any other hypotheses that came out. So co-first author Li Shi-sun at the JCVI led the systematic construction of many strains to find what is the smallest subset of these genes that together are sufficient to restore normal cell division. And this set of 7 genes were together sufficient, and deletion of any one of them, the cell will increase its variation in cell size and shape. But 5 of these genes don't have a known function. They're now implicated as contributing to cell division. But if you look at it, protein of unknown function, DUF177 domain, functional category unknown. So how is it contributing to cell division? So that's motivation for our current studies. So without other hints based on the literature, David Bianchi in the group of Zanluthi Schulten at the University of Illinois has undertaken a study, a bioinformatics study of the entire proteome of Syn3A. And I'll here focus on genes involved in cell division, or at least have a direct role in cell division. Although for people interested in other aspects of physiology, David analyzed the entire proteome of Syn3A. So these are the predicted structures of FTSZ as noted in CEPF, which FTSZ doesn't associate with a membrane on its own. It's possible to modify it for in vitro studies to do that. But inside living cells, typically there are membrane anchors, such as CEPF, which binds the membrane and also binds FTSZ. 520, one of the other 7 genes, is predicted to perhaps have esterase or lipase activity. It's not known how lipase activity relates to cell division, other than hand-wavingly to say that the membrane composition could affect the membrane properties and the shape of the membrane is changing during cell division, but we don't have a clearer idea at this point. 527, I'll discuss that more later in the talk, but it's predicted to be part of a broadly conserved family of proteins that interact with 23S ribosomal RNA. We don't know how that's related to cell division. And these remaining three genes are also predicted to associate with a membrane. 602 is predicted to have two N-terminal transmembrane domains. Consequently, the majority of it should be inside the cell. 604 is predicted to have one N-terminal transmembrane helix, in which case the majority of the protein would be outside the cell. And 605 is predicted to have a lipoprotein anchoring motif. Likewise, the majority of the protein would be displayed outside the cell if that bioinformatic prediction is true. And likewise, we know even less about these proteins. LEMA is also conserved in bacteria, but there's no reported function for LEMA. It's been reported to be involved in pathogenicity in some species, but in terms of its molecular properties, that's unclear. And this bioinformatic analysis also turned up some genes that are known to have direct roles in cell division in other species. There are two. EZRA gene 239 has a structure that can form a large hoop that can enclose an FTSZ filament as well as its membrane anchor in red adjacent to the membrane. And this is thought to somehow hinder interaction between FTSZ filaments that are adjacent to one another and have other roles potentially. And GPSB, gene 353, can also be involved in regulating the assembly of filaments into higher order bundles. And then this can also be involved in affecting the polymerization and depolymerization of FTSZ. So, and furthermore, there are many other genes potentially retained in JCVI since 3.0 that are essential. They're always there, and they may not have a direct role in cell division, but they might affect cell division indirectly. And an ambitious goal is to understand how does cell division emerge from the full set of genes. And of course, the minimal cell offers an exciting model system to ask that question. So that leads to a recent opinion in which we considered other genes in SYN 3.0 that might be relevant to cell division. And we look at this from a physics point of view. What are physical properties that are important for cell division? We focused on two: the surface area to volume ratio, because for a cell to divide, it has to have more surface area than a sphere with the same volume. It has to have enough to divide. And then also the membrane curvature, because of course division involves changes in membrane curvature. And regarding the volume, as the majority of the cytoplasm is likely water, what genes are involved in affecting the flux of water into and out of the cell? What genes are involved in osmoregulation? So we just highlighted that a large fraction of the transporters in SYN3A don't have assigned substrates, but could be involved in osmoregulation. And then as the membrane of mycoplasmas by mass is approximately half lipid and half protein, we considered the small number of genes that may be involved in either incorporating lipid from the growth medium or flipping lipids between leaflets. And there's a variety of ways in which these cells might be generating membrane curvature. One is the cytoskeletal proteins, such as FTSZ that I mentioned. Also, the membrane retains cardiolipin, as shown by the group, by James Sayens's group. Cardiolipin is conical and can localize to curved regions of the membrane and also interact with specific proteins potentially. And then there are also other general mechanisms described in vitro and in other systems. One is that crowding of proteins in the outer leaflet can contribute to membrane curvature. And we also don't know a priori how the energy to deform the membrane relates to the energy to deform the chromosome. Therefore, physical linkages between the chromosome and the membrane could potentially affect the shape of the membrane, but that's speculation at this point. So focusing on our plans to follow up on possible functions for some of these genes, gene 527, why have we annotated it as YLBN, or it has a different name in E. coli, YCED? For one thing, across almost all bacteria and plastids, across almost all bacteria, It's almost always upstream of, to my knowledge, it's always upstream of RPMF, which is a large ribosomal subunit. Furthermore, this gene is also conserved in plastids in plants. And because it's so highly conserved, but has very little known about it, is one reason why we're interested in studying this gene, which has a phenotype in the minimal cell. Furthermore, in David Bianchi's work, the predicted structure for 527 is very similar to the predicted structure of wild BN and Bacillus subtilis. So that's why it's been annotated as this gene. And there's really only one paper that we're aware of in which they showed that in an E. coli strain lacking this gene, there's a significant reduction in the amount of 23S ribosomal RNA. But they showed qualitatively there's no effect on growth in that when they streaked the cells out on plates, there wasn't a major change in the density of cells that were observed. But to my knowledge, other phenotypic changes weren't investigated here. So as a starting point, we're planning to investigate in an in vitro system whether purified 527 interacts with 23S ribosomal RNA as a starting point. And then as a last gene that we're considering now is when we published this opinion in 2022, we speculated there might be a gene that is involved in uptake of lipids because if mycoplasmas are treated with protease, that can affect the uptake of lipids. And it also makes sense that a cell which depends on lipids from the growth medium would have some genes perhaps that facilitate that. And just a few months ago, a paper was published by another group. I had nothing to do with this work, describing a protein in Mycoplasma pneumoniae, which has a function involved in incorporating lipids from the growth medium. It's distinguished by a large hydrophobic cavity that catches lipids. And then by a mechanism that's unclear, it incorporates those from this large hydrophobic cavity to the membrane. So this is now the cryo-EM structure of that gene from Mycoplasma pneumoniae, the hydrophobic cavity shown in green, alongside the predicted structure of gene 33 from David Bianchi's work. And David Goodsell in his recent paper also described a large protein distinguished by a large hydrophobic cavity. And one thing that's exciting is that there are 5 lipid types in the Syn3A membrane. In 2 of them, genes are retained involved in their synthesis. And then this protein in the functional study by the other group led by Sprankel et al. Has specificity for the other 3. So in a sense, if this gene does have this function, in a sense, completes the input to the lipid metabolism of Syn3A. And we're interested in it from the point of view of what sets the growth rate of the membrane for surface area to volume ratio. But of course, it's relevant to other group. And then the Science group is doing amazing work on the lipid composition and metabolism. So with that, I would like to thank you all very much. And thank all those who contributed, and we appreciate your questions. Thanks again.
I know I'm closely related to this work, but I'm always dazzled by it, James.
Oh, thanks, John.
Questions?
I know you sent this paper before, and thank you for doing it. And I think soon it would be awesome to connect again, but I think we'll take longer than yourself, the scope please? Of one question in this talk, because I have a lot of things I am thinking about and would like to ask, but I'll hold off until we have the proper time.
Sounds great. This is Isaac Justice. This is Isaac Justice.
Very exciting work.
Isaac, okay. James, if you could know one thing that would help you more make real progress proving or disproving your model, what would it be?
Well, I think that the lipoproteins to me seem super interesting because I mentioned this hypothesis based on in vitro studies that crowding of lipoproteins can generate membrane curvature, and membrane curvature can cause cells to constrict, fully constrict, and even divide in vitro studies. And in those vitro studies, the density of lipoproteins was roughly 10-fold lower than the expected density of lipoproteins based on proteomic estimates of the abundance of the lipoproteins and then estimates for the size of the cell. So if we knew, how many of each lipoprotein were there and what was their density, the question could, in a sense, get flipped on its head and say, why don't cells divide at very small sizes? Because the crowding of lipoproteins would be so high that it's just driving such a high degree of curvature. So I think, yeah, just as a general question of what kind of information would be really helpful, would just be better characterizing those lipoproteins, because I think many of them are described as uncharacterized lipoproteins. I'm sure they have specific roles, especially in the environment of a host where they have to evade the host immune response. But of the lipoproteins that remain, some of which are essential, what roles do they have? Are they, probably some involved in transport? But I think, what effect do they have on cell shape? I'm really curious about that. Yeah.
And keep in mind that the quantitative proteomics estimates that we use, the proteomics guys have always said that the membrane proteins take those numbers with a grain of salt. They're pretty confident about the cytoplasmic proteins. So who knows? Elizabeth, you look like you have a question.
I do. Elizabeth Strahalski at NIST. Hi, James. Great talk. Thank you.
Thanks, Elizabeth.
So you've been talking about how there might be spontaneous cell division based on membrane curvature, but I'm actually wondering if you can comment, now that there's been all this work since the original observation of these strange morphotypes of SYN 3.0, do we have any more insight into what might be stabilizing some of those strange filamentous shapes and other just weird cell shapes, which aren't spontaneously dividing from each other?
Thank you very much. Thank you, Elizabeth. Related to that point, so for a cell to fully constrict, that's predicted within these spontaneous constriction models, but there's a predicted energy barrier from going from being fully constricted to being fully separated, involved with, one way of thinking about it is, for a cell to change its topology and divide, it has to open a hole in the membrane, which would then have to expose that hydrophobic core of the membrane to solvent, which is energetically unfavorable. So one way of thinking about it there might just be a thermodynamic block on going from fully constricted to divided. But, if that's the case, what is it that generates, the force, say, for a cell to then be able to go from fully constricted to divided? I think that we know that it is possible in the absence of external forces based on our microfluidic experiments. I didn't show these, but in Elizabeth's lab, we conducted experiments imaging the growth of cells in chambers where they're not exposed to flows. And in these chambers, we didn't quantify this, but qualitatively small cells are generated that are disconnected. So the cells don't need to be pulled by external forces to divide. And then one last thing I would note is, in walled bacteria, where FTSZ contractile ring is a paradigm for cell division, there's reports that when the cell approaches an inner diameter of hundreds of nanometers, FTSZ dissociates. In other words, and also, if you think about it in terms of how much space does the FTSZ ring occupy, at a certain point it has to dissociate before the cell divides. So it's not that FTSZ can fully divide the cell and then maybe separate it. Maybe it does. I don't know. I'm speculating at this point, but yeah, I think to your point, what is it that goes from fully constricted to then fully separated? I think that's unclear. And then, as far as if there's a specific gene stabilizing, I don't know, but that's a really interesting possibility that you raise with the question. And of course, if you or John or anybody else has other thoughts, that could be really, yeah, please. But thanks for the question too.
James, you have shown in the Cell paper and in numerous talks SYN 3.0 in a microfluidic chamber that appears to be a long tube as opposed to a spherical cell. Can you imagine a long tube forming without some cytoskeletal structure to make that happen?
So a really cool thing is that some models of vesicles do predict large, long tubes. And furthermore, in vitro experiments, long tubes are sometimes observed. So Membranes can form tubes in the absence of cytoskeletal filaments. The question becomes, what's the diameter of that tube? And relate to Elizabeth's question, how stable is the tube? And I think, yeah, just as a general idea, it might be that either the tube formation or cell division might be thermodynamically favorable, and then the specific genes acting within the cell might somehow regulate that process or speed up the process, but they might not be necessary for it to happen at all. I think that's a bit hand wavy, but some of the membrane tubes I've seen in literature are tens of nanometers in diameter. If SYN3O is forming a membrane tube that's hundreds of nanometers in diameter, I don't know what the requirements for that would be, or if something else would be required to generate that. So, yeah, I appreciate the question.